wear my hair, my hat really low. Yeah, I know I'm not my fanciest today, everyone. I don't really apologize that much, but like I'm gonna go kayaking after this, so um yeah, I mean, I know. I don't generally like dress up to do that. So I've like actually got like just a tank top and a bathing suit on and pants over that. So, and no makeup, obviously, and a hat and sunscreen already on. I'm like literally ready to go. So I'm gonna go through this. It's just me right now. That is fine. I fully expected that on a Saturday morning. So no worries, I'm not judging. And I like to have something distracting on in the background just because otherwise it's like literally this big blank space in my office. You know how I am. You guys already know this about me. Like I like to have either music on or something. So there's no sound with this, but it is something distracting. It's mostly just for those of you who play online games, this is mostly just this is in Sim. He's a gamer and he's playing video games in the background of me lecturing you guys about bacteria. So I don't know. I watch a lot of Let's Plays. So for those of you that don't know, other people playing video games. I don't have the mental capacity after talking about bacteria and viruses all day to come home and play video games. So I watch other people play them because that's how exciting my life is. So we're gonna get into bacteria today. And this is mostly gonna be me kind of just going through concepts for chapter 4 that are on our study sheet. So it's stuff we should already know, but just kind of a refresher of that. So I'm going to run through that information first. And at the end of this, so for those of you who don't care about the rundown, or for those of you who don't care about the end of it, you can decide which part of it you want to watch. But this first part is going to be me running down the information that we should know about bacteria from the study sheet. And then after I get through that, I will be going through some open-ended questions. I mean, they are actually literally critical thinking questions, but not the same kind that you would see on the NCLEX. They're just like open-ended questions that you have to think about in order to answer them. So we'll go through those. They're not multiple choice. Like I said, those are going to be open-ended. And I did make a PowerPoint for those. There's only 5 questions, but still, we're going to go through them anyways. And I will post those. Hopefully I'll remember to do it this time, but post those with that onto the announcement board when I post this video. Because again, I don't anticipate anybody joining at 10 o'clock in the morning on Zoom to do a review. So I know it's probably going to be mean, but people don't care that much about bacteria, but when you guys come back to study later, you're probably going to care. So I wish that you watched it for whatever your reason might be. So starting out with chapter 4. Chapter 4 covered that concept of bacteria. But I also just want to remind you guys of one more thing that chapter 4 covers that we like almost never talk about again. We mention it very briefly here or there when we're talking about the different environments that bacteria can live in and stuff like that. But we're going to mention them again in the GI disease chapter in chapter 23. Whenever we do get to chapter 23 in unit 4, they're going to come up again. And that is the fact that chapter 4 isn't just about, why is this not wanting to write? I don't know. Well, that is, I don't I don't know. I don't know what's going on here. This is me using the eraser, by the way. So apparently I'm not going to be able to be using this whiteboard like I thought I was. It's literally not working. So I apologize for that. Eraser, it's not working. Not with, I'm using my mouse now. Like usually I use this thing, but you guys know that the board or whatever it is, it's not doing anything. Mm, mm, mm. or whatever. But it's archaea. They won't let me write either. That's great. Oh, there's that. Like, this is what I'm not understanding. But it won't let me erase. No, it's only like, get this. Let me try a new page. It's showing it erased on this. That's weird. I don't know. I don't know. I don't get it. Nope. Okay, well, we're not gonna be able to do that then, I guess. That's incredibly frustrating. Like, I'm not clicking when it does this. I want to be clear, like it is just like moving my mouse around and I can't control it. Like, I can write with my mouse, but I can't actually write with my mouse. That's the problem. And then when I go to erase things, it's like selective. It's not actually like it erased those things, but not all of it. I don't know. It's really weird. Anyway, I don't know. So I can use this. Like I didn't know it was called. Those are about to judge me because about to do something I do when I'm angry, which is trying to. What I get, I guess, for trying to get up early and help people bacteria. Okay, so the other one's archaea, right? And so we know that archaea being you know, the both bacteria and archaea, the big feature that for some reason people just could never remember is the simple fact they do not have any membrane bound structures in the cell. So they completely, well, you can tell I'm frustrated. I don't know how to remove that. I apologize. I have no clue how to remove that weird arrow. Here it is. Okay. All right, so they completely lack any membrane bound structures in the cell. And that's important. Like everything in the cell, everything inside of their cells is contained by what we call the cytoplasmic membrane. It's contained by the cytoplasmic membrane, or, and I say or the, and I don't mean this is another completely different structure. So let me rephrase this actually. AKA the cytoplasmic membrane. We can also just call it the cell membrane. AKA the inner membrane. In the case of the organisms, why? Which have more than one membrane. which we'll get to in a minute. You don't have to, I know you guys should know that's gram-negative, right? Gram-negative have an inside membrane and an outside membrane. So you should be immediately thinking that whenever I'm reviewing this, but we are going to get to that. That's part of this chapter. And so we will actually be touching on that kind of like, I won't say separately, but it gets its kind of own topic that we're going to get into. Okay, so bacteria and archaea, regardless of what differences they may have genetically, they're actually completely different genetically, like not 100% different, obviously, because nothing is 100% different. I want to make that clear. Even bacteria from us, nowhere near 100% different from us. We share genes with them. Yes, we do. And our genetic codes and everything like that, there's so many similarities there. It's not like they're 100% different, right? And that goes from bacteria to archaea as well. They're not some sort of weird alien or anything like that. They share genetic code. When I say genetic code too, really, I am also just referring to the fact that they use A, T, C, and G. They use nucleotides. They have DNA, truly. They have RNA, truly. They have ribosomes, truly, they do. So the fact that they have all of those things together, that's what makes them cells, right? And so the predictability of that, the way that it works, the way that it operates, that's how life as we know it works. So they have to have those things in common. We already know that. So that's what I'm talking about when I say that they do have similarities. Now, what do they look the same under a microscope? I mean, at face value, archaea and bacteria look pretty similar, but that's like, they really are not very similar beyond that. Their genetics are more similar to archaea has similar genetics to us, more so than they do to bacteria. They also have ribosomes that are more similar to ours than they are to bacteria. So that's actually pretty impressive, even though we're lumping them in here with the prokaryotes or the eukaryotes. So that's another word we can bring up here. We can remind you of the word eukaryote. And what eukaryote means is that there is no true nucleus. So these guys lack a true nucleus. And what do I mean by a true nucleus? A true nucleus is a membrane-bound structure that encloses the genetic material of the organism. So these guys are eukaryotes. That's literally what the word means. No nucleus, without a nucleus, okay? So they are eukaryotes. I don't know, this is why there is a motion with my front door. I don't know. Anyways, so eukaryotes don't have any true nucleus. What else do they not have? Like it says at the beginning, at the very, very top there, they completely lack any membrane-bound structures. So when we get into the eukaryotes, I'll remind you about the membrane-bound structures that can exist within eukaryotic cells. Remember, eu being true and karyote being nucleus. They do have true nucleuses, the eukaryotes do. But these eukaryotes don't. They lack any membrane-bound structures. And membrane-bound structures will include things like mitochondria and endoplasmic reticulum. Any sort of set structure that's divided that way, they don't have anything like that. 0% chance of it ever happening. So if you ever have a test question that asks you about the mitochondria, how mitochondria work within a bacteria, that is a trick question, 100%. Okay. Bacteria don't have any membrane-bound organelles ever in any bacterial strain or species ever. So don't ever think that they're going to ever have anything like that. So if there's ever a question like that, is automatically they don't have that, though. That's a trick question. So think of it that way. Okay. No membrane structures at all, and that includes the nucleus. So where is the genetic material of a eukaryote? If they don't have a nucleus, though, where is their genetic material? The genetic material of bacteria and archaea is found in the cytoplasm of the cell. And I'm gonna put another little comment here, which is to remind you guys of cytoplasm is the water-based, let's say jelly-like content contained within the insides of a cell. So everything inside of a cell is not just sitting there by itself. It's floating within something, this jelly-like fluid called the cytoplasm. The cytoplasm is like 95% water. So that's why you yourself are made up of so much water. It's completely because of the cytoplasm. The walls, or say the walls of the bacteria, or the walls of the plants, or the walls of fungi, or the cell membranes, or the membranes that surround the endoplasmic reticulum, or the ribosomes themselves, or your DNA, or your RNA, any of these things, that's not water, right? I mean, we can agree on this. Those things themselves are not water, and they are never made of water. A wall is made of what, for bacteria, the peptidoglycan and all of that, like that's not water. So whenever we're talking about cells being made so much of water, and the substance of living things being made so much of water, then clearly that water makeup of these organisms isn't going to be based on those structures in the cell. Where's the water come from? It comes from the cytoplasm. And the filling, the space in between is all water-based. So that's what we're talking about when we're talking about the water there. So yeah. That's what cytoplasm is. That's a term. This is a term. I'm going to underline this one so that we can see it more. Well, maybe not, I guess. But let me do bolding, but it didn't let me do underlining for some reason. Bold it. Okay. So pretty good there. So all of our genetic material, if we are a bacterium or a archaea, all of our material is hanging out in the cytoplasm. And more specifically, it's not just in the cytoplasm. I'm so sorry, my nose is itching so bad. Not just in the cytoplasm, but it is also hanging out more specifically in this area. Sorry, I have to type and think at the same time. So it's located in a region of the cell in the cytoplasm, in a region of that cytoplasm called the nucleoid region. That is the area where we can actually find the genetic material of our bacteria. Okay, so cool. So let me pull something up real quick. A small one. All right. So that's that part. I wish I could delete this. This is so annoying. This is here. Moving on. So both archaea and bacteria no membrane-bound structures, they don't have a nucleus. So where do bacteria and archaea keep their genetic information? In the nucleoid region of the cell, and that's just an area, really, where it's not an area that exists separately outside of the existence of the genetic material. It exists solely because that's where the genetic material is located. Okay, it doesn't exist. If you take away the genetic material, there is no nucleoid region. It exists because that's where the genetic material is. So that's what that is. So if I ask, where the genetic material is, you would say the nucleoid region. I would say, well, so what is the nucleoid region? Describe that to me. Where is that in the cell? You would say, well, it's just the area where the genetic material is. That's literally all it is. If there's no genetic material, there is no nucleoid region. It's just describing where it is. So that's all that is. Don't think it's a separate thing that exists outside of that situation. So that's the definition of what bacteria and archaea are. They are eukaryotes because they lack a true nucleus, and any other membrane-bound structures like mitochondria or the endoplasmic reticulum or the Golgi apparatus or whatever. But just don't forget that nucleus falls within the realm of that understanding of what a membrane-bound structure is. The nucleoid region where we find the genetic material of bacteria and archaea is not membrane-bound. Okay, so cool. So that's where we are in this. So let's move on to next things. So the next parts that I have, so this figure is over here, so you guys can refer to it as we're going through the different parts and functions of the cell. I'll probably have to copy paste it as we're moving through it, but since we're typing things, I want to have something that you can refer to. And apparently, if I touch some sort of aspect of this text box, that can point to certain things. Like if I'm here talking about the nucleoid region, and that's really kind of what we've talked about here, that this would be where our genetic information is right here. That purpley massive mess, that is our nucleoid region, and where we would be having our nucleic acid hanging out. Okay, so yeah, it says bacterial chromosome or nucleoid. Yeah, nucleoid region, that area where you're finding that genetic information. Now, why isn't it just dispersed randomly all throughout the cell? Like, why isn't it just like parts of it here and parts of it there and little bits of it here and little bits of it there? The genetic information of the bacterial cell isn't dispersed randomly in parts all over the place because there's not more than one chromosome. There's typically, in most bacterial cells, typically only one chromosome, and it's circular. It is DNA, just like we think of any other cell. All cells have genetic material that's made of DNA only. So viruses, when we talk about their genetic material being potentially made of like single-stranded RNA, that's not a cell, so it doesn't fall into that category, right? All cells, all living cells have DNA for their genetic material. And same thing with bacteria, they're living cells, they have chromosomes. So that's what their genetic material is made of, is DNA in chromosome form. Almost, I don't say almost, that's not, I don't want to say that way, but most bacteria have just one chromosome. It is circular. Most of them are going to just be one circular chromosome. And so you might be thinking or saying to yourself, hey, but let me just point something out really quick, that when we look at, I'm going to try to do this again, it's probably going to draw a weird shape or whatever. When we look down here, we've got this weird circle down here. Isn't that the genome? Isn't that part of the chromosome? Is that not the bacterial chromosome? It is not the bacterial chromosome. That's the plasmid. So not all bacteria have plasmids, but when they have plasmids are extra circular bits of DNA, okay, that some bacteria have that carry extra genes that may confer antibiotic resistance or bacterial toxins or whatever thing like that. And they can be transferred between bacterial cells in some cases, or picked up from outside of the cell, or transferred by genetic engineering methods as well, which we'll talk about in the review for chapter 6 and 8 together. So think about that way. But anyways, moving on, that chromosome is all in one, it's one piece. So it's not going to be in pieces all over the place, because it's not like our chromosomes where we have 46 chromosomes. that if we didn't have a nucleus containing them, those chromosomes could be scattered randomly throughout the cell. So that's not what's going to be happening here. So, but yeah, that's why they have a nucleoid region and why they have a chromosome, not more than one. So, okay, so, all right, so let's move on. We can have in bacteria, we learned next about the cell shapes and ranges. So I'm going through the study sheet itself if you guys are following along. Then I got stuck up kind of and they don't have membrane structures and it went off. But now we're moving on to cell arrangements and shapes. And so here are the ones that we learned about. We learned about the cocci, which are round. We learned about the bacilli, which are rod-shaped. And then we learned about pleomorphic cell shapes, which that just means that you can have no set shape of your cell for that species. So like if you were to look at mycoplasma, they don't have any cell walls. So it's hard for them to maintain their shape so much. There's still some rigidity in their cell membrane. They have added cholesterol in their sterols help to give rigidity to membranes and to help hold the shape of the cell, but it's still not super effective. So when you look at them under the microscope, even though you're looking at all a pure culture and they're all the same species, they can have all different shapes of cells there because of that lack of ability to basically maintain cell shape. So there's that. But yes, so let's look at this. And then let's do this, let's go to arrangements. Okay, and then for our cell arrangements, the ones that we mentioned in the study sheet are going to be ones that we should all be familiar with most of this stuff. This is my good guide. This is going to be Staphylo. So this is going to be irregular clusters. Next we have, I'm sorry, so I'm having time keeping track where I am on this. Strepto, which is going to be chains. And when I say chains, chains should look like this. Okay, so if you guys see something that looks like here, I wish that this was bigger. I tried to draw those like this. Here, and then here, and then here, and then here, and whatever, and like this, and like this, and like that. And you could say like, oh, but it looks like they're chains because of how they're all stuck together like that, and whatever. I'm like, that looks more like irregular clusters to me. That doesn't look like a chain. I should probably break this news to you when we're in actual lecture or we're in actual lab, and I promise I will, but some of you guys are describing your cell arrangements as strepto. There are some of our bacilli, and when I say bacilli, I mean they are some of our very long bacilli, like in the gram positives that usually are on there's one species that makes strepto arrangements like end to end to end to end to end linkages in like a chain like fashion, like we have the O's in the picture down there, the text. Anyways, there's like one rod shaped bacteria in the gram positives that does that. And like I have a lot of people who are saying that their organisms are strepto. And I'm just wondering if you guys think, if you really understand what strepto means, because I think there might be a misunderstanding there. So we'll have to discuss that before the next exam. which is, it's fine, but, I just wanna make sure that you guys are clear on that. It's a chain, they're linked together, they're not gonna be in some clumps and then a couple like of them linked together like a chain, like 3 cells, and then there's clumps over here. When we're talking about the arrangement of your cells when you're entering into your unknown report, we're talking about the characteristic arrangement of your cells. The one characteristic arrangement of your cells, not that you looked in your microscope one day and saw 3 of them that were linked together in a chain, but everything else was sort of random. To me, that would be, oh, okay, then your characteristic arrangement of your cells would be random and not an arrangement, which you don't have to have an arrangement. Sometimes you have arrangements, and that can be a characteristic defining factor of a species. And that's why we want to document that when you're looking at it, but not all of them are staphylo, not or strepto or whatever, sarcina or whatever else it could be. They don't always have that. These are just possible ones when you do, okay? So don't get so obsessive about it has to be staphylo or strepto or palisades. Like it's, and for some reason you never, nobody ever sees that one, but they always see strepto. You know, it's not there. It's not there. Okay. So, well, you guys maybe look at it a little bit differently. I think what I'll have to do, well, just me talking to myself, doesn't matter, you don't care about that, but I will have people point it out to me next time. You're like, oh, you think it's strepto? Show me. Show me why you think that is. Okay. So we'll have to do that. Maybe not put you guys too much on the spot with it, but next semester is going to get that one. All right. Then next we have palisades, which I just mentioned it, but this is going to be like fence post type arrangement, like fence slats. So upright next to one another. So instead of chained out in big long chains end to end. So instead of it being like those are minuses, so that would be like the lengthwise, like bacilli next to each other. Instead of it being like that, it would be like this. Side by side. And this is only in bacilli. Okay, and then our next one was tetrads. This is just groups of 4. And then we also have on here sarcina, which are cuboid, literally just cubes. So now instead of just group of 4, now we've got a true cube, like a 3-dimensional cube. So a 4 cubic. You have like 1, 2 this way, 1, 2 that way. So that would be already. So if you have, let me just, I'm not describing it very well. I know you guys know what I'm talking about, but still I want to try to describe it better. So if you had this, that is a tetrad, right? But I can't type it out on this because of how just that's how 3D works. But if I were to try to draw it, you would have a 1 and a 2 and a 3 and 4. This would be a tetrad, right? And then sarcina, you would have back behind this guy, this one, and then back behind that guy, that one, and then back behind that guy, that one, and then back behind that guy, that one. So if you know how you would draw this out 3D wise, you would have this like connecting of all of these squares. And then you'd have that sort of 3-dimensional shape, just like you would get if you were to draw it out. So that would be like that 3-dimensional look at just the corners of that. So that's sarcina. It's a cube, right? Whereas tetrad would be just the 4. So it's like a 2-dimensional instead of sarcina being like the 3-dimensional version. Okay. All right. I'm frustrated trying to drive me right now because I'm trying to quit smoking, but the better way to deal with that tends to be vaping. No, zero nicotine, but only certain things will give you comfort. Unfortunately, we all have our vices. Better this than smoking cigarettes, honestly. But okay. So page. Move this onto this page. Okay. All right, so we talked about cell shapes, we talked about arrangements. Let's move on. So other features of bacteria, capsules. So actually both of these, whether we're talking about capsules or we're talking about slime layers, these are the glycocalyx. This is the term for it. So capsules themselves are more tightly associated, and therefore adherence to surfaces and for phagocyte evasion, because they make it, the capsule makes it harder to phagocytose or eat the target encapsulated bacterial cell. Okay. Yeah. I wish this would have dialed. So used to it and everything else that I do. Okay, so that is the glycocalyx. Capsules, and we also have the slime layer. And this one is more loosely associated, and it's for attachment more often in biofilms. And remember, a biofilm is when bacteria of one or more species are attached to a surface and then to one another as they build and can respond and react to the chemical signals from waste, etc., of nearby bacteria that incorporated in the biofilm in order to maintain an understanding of the overall health of the biofilm as a whole. And this will include things like plaque or biofilms that grow on implanted medical devices. Okay, so that is biofilms. Just so we're all on the same page. So any of the glycocalyx can have to deal with attachment. The capsule is involved more in evading the phagocytic aspect of things than the slime layer is. And both of them can be involved in forming biofilms. So, but we usually think more of outside of like the body. Typically, we're going to think of slime layers being involved in biofilms, like the biofilms might be forming in like palm scum or on surfaces of rocks or something like that out in like nature. But in the body, it could be the capsule that's associated with that. So, which depends on what you're looking at, but both of them can be associated with that. Glycocalyx, okay. I know this talks about it. It's this like kind of fluffy layer or whatever here. All right, next we have the flagella. They are motility structures. Let's just say they move in a rotational fashion. So either clockwise or counter. And the switching between the directions of rotation of the flagellum, which is the singular, is what dictates direction of movement of the cell. This results in runs and tumbles. Runs occur when the rotation is just smooth and unstopped. Tumbles happen in the direction of the rotation. Switches. Okay, and then the bacterial cells will make use of the switch to direct their movements. And then bacteria can move toward or away from certain stimuli that are considered favorable or unfavorable. So like smelling cookies or smelling smoke, right? Smelling cookies, favorable, you move towards that. Smelling smoke, not favorable, not to your survival, right? So you move away from that. So same thing with the bacteria. They are judging things based on that. And they will use signals that are chemically based, which are called chemotaxis, or light-based, which is photosis. So movement based on chemical signals or movement based on light. And you might be more concerned about the light signals. If you are an organism, that is a photosynthetic organism, of course, right? So that's how the flagella and the flagellar movement would be working. So here's the flagellum here. Let's see. Sorry about the spelling. I don't look at the screen while I'm typing. If you get me in a groove, then I'll look at the screen. But you have to give me in a groove. Otherwise, I'm gonna look at the letters. Sorry. I'm good enough at typing to watch the screen while I type, but I am just not in that mood right today. In the mood of kayaking. So, right, so that's good there. So our next topic we have here are Fimbriae and Pilli. So Fimbriae are attachment appendages of bacteria, and they are not used for movement. And I will add in here only flagella are used for movement in bacteria and archaea. Okay. Does this talk about a pilot? Yes, it does. All right, perfect. Okay, the pilus or the sex pilus. And you know what the bacterial cell is using it for in that particular moment. This is used for, I think, some cases attachment, but mostly this is used to transfer genetic information from one bacterial cell to another of the same species. This even include information relating to toxin production or even antibiotic existence, and can transfer small fragments of genetic info or even in that's actually fine. Right, so I talked about flagella. When I guess we can come back over to this flagella stuff because I didn't realize I had organized it this way in my study sheet. My bad. Well, let's go ahead and go over the arrangements of the flagella. So the different arrangements of the flagella. You can have a bacterial cell. This is going to be, you can have just one. All right, you could have flagella at one end and another end. So this could be single at each end or group. Either way, it's like having this end and this end, right? Having it at both ends. You could have a group at just one end. So not one at just one end, but a group now at just one end. Or you could be covered all over. So if we're talking about just one at one end or the other, right? This would be called monotrichous. If you have them at both ends, this would be called amphi. Remember amphi, amphibian, both things can be happening. So amphitrichous, you could have flagella at both ends. You could have a group just gathered out at just one end. So this would be one end is lophotrichous. I don't know why, but it's easier for me to remember lophotrichous when I think of it not as a group at one end, but a tuft. I don't know why, but that word just resonates with me more that way. And if they're covered all over, this is called peritrichous. So that's the flagella. So I'm actually going to go back to slide number 1 just really quick to touch on something because we're talking about lacking membrane bound structures and all of this sort of stuff. So I'm going to come back to this. Let me one more. Okay, so we're talking about bacteria and archaea and the general idea of what they are. So let's talk about the cell envelope of these guys. The cell envelope. Okay, well, we're just going to stick with bold then. Consists of the membrane that directly contains the cytoplasm and all of the rest contents of cell. Perfect. And that membrane is called the inner membrane or the cytoplasmic membrane or the cell membrane. They're all the same thing. Okay. So it includes that, the inner membrane, the cytoplasmic membrane, plus any cell wall that might be on the outside of the cell. So if we're talking about cell wall, I'm going to come back to that one in a minute, actually. So just and then plus any outer membrane, which we see in gram-negative bacteria only. Okay, so I want to talk about the outer membrane. Because gram-positives have that thick cell wall, they don't need an outer membrane ever. Okay, so that's immediately always 100% of the time if I'm talking about gram-positive versus gram-negative to you guys in class or in lab, anything like that. If I'm talking about gram-positive, they always have a thick cell wall, always, and do not have an outer membrane. If we're talking about gram-negative bacteria, they always have a thin cell wall if they have a cell wall. Now, the ones we talk about in our lab, when we're doing our gram stains and everything, we aren't discussing the fact that they might not even have a cell wall, because the ones that we're testing always do have some kind of a cell wall, but it's just so thin, it's not even holding on to that crystal violet stain when we're doing our gram stains, right? But because that cell wall is so thin, they also need that extra protective layer of that outer membrane. Only the gram-negatives have that, okay? So, so that's what you need to know about gram-positive versus gram-negative. So that's the cell envelope. It is always that inner membrane. Every single cell ever in the history of cells has a cytoplasmic membrane, okay? They always have at least one membrane on the outside containing everything. That's how the stuff's in the cell. Then we have a cell wall most of the time when we're bacteria, right? Then we have an outer membrane if we're gram-negative, because we need that extra membrane to keep it all together. So that's the whole of the cell envelope. So the cell envelope is not the same thing as the cell membrane or the cell wall. It's all of that outer outside all together contained. All right. Okay. All right, so now let's break it down. What is happening? Okay, so now let's break it down from this differences in our cell walls, all right? Okay, sorry, I email, I promise I'm getting back to it in just a second. All right. Gram-positive. We know that we can divide organisms up based on whether they are gram-positive or gram-negative. Easy, we already know that. So gram-positive, these guys have that thick cell wall. And the gram-negatives have the thin cell wall. Great. So that's the biggest difference right there. These guys are so thick on their cell wall, the gram-positives, that it traps the still violet in the gram staining procedure. Okay, perfect. All right, what do I say about this? So they both have inner membranes, yes. The gram-positive, we are surrounded by that thick cell wall. The gram-negative, we have a thin cell wall, but that is surrounded by the outer membrane. So let's see this. So it says any cell wall. The cell wall is made. Let's say, actually, we'll specify here. Bacterial cell walls, okay, is made of peptidoglycan. Repeating sugar units linked together peptide cross-linking bridges. Peptido, peptide, right? So if we had peptido, peptido and glycan. So that'd be peptide, peptido, like in protein peptide group, amino acids that are linked together. We already know peptide, excuse me, proteins. What are proteins? Amino acids linked together in a specific sequence, right? Each amino acid differs from one another from that R group that's on the outside of it. That's hanging off as like its little chain group, right? And that determines what each amino acid is, whether it's lysine, whether it's a proline or glutamates or whatever it is. It's all determined by that little R group, which is R because that's the variable with the group that it can have. So they're all linked together, those amino acids linked together by a bond called a peptide bond. So when I say a peptide, I'm talking about a group of amino acids that are linked together by peptide bonds. And when they're in short groups like this, they're just called peptides. They're not proteins. They themselves do not have a function separately. This is just a bridge that's connecting strings of sugars together. And that's all it does. It's not a protein separately from us, any other function whatsoever. That's all it does is act as a bridge for this scaffold building of the peptidoglycan. So peptido, that's the amino acid, short amino acid sections, peptides. Glycan, you want to say glyco or glyco sounds like a glucose, sounds like that's like sugar. Yes, you're correct. That's absolutely right. Glycan is referring to those sugar groups that are linked together. So the repeating sugar links, repeating sugar units, excuse me, that's the glycan part linked together by these cross-linking bridges. Those are the peptides. Okay, so peptido, glycan, and really can be sugars. Okay, so sugar. Let's do that. Right, to help you guys remember what it is and how they're made of here. So that is what a bacterial cell wall is made up of. It's either a very thin layer, maybe even just one layer of that peptidoglycan, sugars linked by peptides, or a very thick layer. If it's thin, that's what we see in the gram negatives. If it's very thick, that's what we see in the gram positives. Let's see. Oops. Of course, because why wouldn't you do it that way? So that you guys can remember which one's rich. Which one's which, not rich. Okay. It's not my daily name. These are purple in the gram stain, and these are going to be red because of the safranin counter stain. All righty. Moving on, let's see, we have the acid-fast bacteria and the mycoplasma. So using the acid-fast stain, which in our case, using Kenyon's method of the cold staining method, the carbon fusion, which was that pink, bright pink purple color. If that color stays because it is lipid soluble, that means it's binding to that waxy substance that's on the outside of the gram-positive cell wall that we find in acid-fast bacteria, right? So they say acid-fast positive. I don't know how to spell fuchsia, so please don't judge me. We know what it's supposed to look like, right? So fuchsia, whether I put the C or the S, I think it's this. Yeah, I was right. Okay. One of those words that like I almost always get it right when I spell it, but then it doesn't feel like you're spelling it right when you write it, you know? Anyways, fuchsia, whenever we do the acid-fast stain. So these guys are acid-fast positive because lipid soluble, and this is me bringing the lab and the lecture together because it's that time, right? So everything is going to be, it's more helpful to have it all together because the more it makes sense in your mind, the more it's going to make sense when you're trying to answer questions about it on the test. Are you going to see acid fast again in the lab? I mean, we're in a whole other part of the lab where we're looking at inoculated media and looking at color changes of the media. Are you going to see acid fast again? I mean, on the test or anything? Yes, it's a comprehensive test, so you will see it again. Get ready. There were quite a lot of questions about it on the midterm test, right? So you're definitely going to have at least one. I didn't put that many questions on the midterm because I thought it was a waste of your time. It's because this is the type of test you're actually going to interact with in a hospital setting. So just be aware of that this is something that if you do a round in an ER, which you will, right? Especially as you're working on becoming nurses, you're going to work in all these other areas of the hospitals, even if you don't plan on staying there. So if you are a nursing student, then that is something that you will encounter. If you're a vet student, then I'm sorry that you have to sit through it, but that's part of the whole routine, right? So yeah, but this is easy now, right? You guys know this. It's so easy. So, the acid fast stain, it's going to have these lipid soluble stain molecules of the carbonation binds to the waxy or lipid-based. Wax is a lipid. Wax is a lipid. It doesn't mix well with water, right? It's a lipid. So wax, waxy, that waxiness. It is lipid-based. All right. So that's mycolic acid. That's the molecule that's waxy on the surface of our gram-positive acid-fast bacteria. So this is on the surface of the cell long mycobacterium species. That's SPP means species. So we're talking about multiple species within Mycobacterium genus, okay? So I'm going to bold this one. So it's the presence of that mycolic acid that causes the carbonyl fusion to stick to it, even in the presence of that acid rinse that we do, okay? Normal bacteria, you do that acid rinse, it rinses away the carbonyl fusion. There's no lipid on the outside of that to hold it there. So that waxiness, it's what makes them so hardy. The mycobacterium, like tuberculosis, makes them so hardy. So they're hard to get rid of off of certain surfaces. So I'm gonna go ahead and. Why is it ringing? I'm so sorry, I'm not trying to do this, but it's like somebody was calling me, but I wasn't actually getting like. It wasn't ringing. Weird, I don't know, that was really weird. Anyways. Okay. So this is gonna include pathogens that are medically relevant, right? Like Mycobacterium, tuberculosis. Sorry, I stopped reading all that. Tuberculosis and Mycobacterium leprae. And there's other ones, but these are the more obvious ones. So obviously we're talking about here tuberculosis and leprosy. There's also like Mycobacterium avium, which produces tuberculosis in AIDS patients. So that might be considered a medically relevant one as well, especially if you are going to be working in an ER where you get homeless folks and they happen to have a pretty high population of HIV positive AIDS patients, or they're being affected by AIDS because their HIV isn't being controlled. So that's something to be aware of in that regard. Pretty happy with that. And the other one was mycoplasma. So we've got our cell envelope regarding our gram-positive and our gram-negative with regard to the wall. And so another one that falls kind of under that gram-negative category because there is no cell wall is the mycoplasma. These guys don't have any cell wall, and they therefore have a pleomorphic cell shape. And they have their rigidity. Rigidity or any shape at all of their cells is maintained by sterols that are present in the cells, in the membranes. That's what gives our own cells their shape and their ability to have some kind of shape, as opposed to just being super liquidy and no shape or whatever. But we're still quite soft. We don't have the same rigidity as like bark, on a tree. So mycoplasma, they do not have a cell wall. They have a pleomorphic shape because of that, but they do have some rigidity added into their shape due to sorry, brain fart, I know. Some rigidity added into their shape due to the sterols that are present, like cholesterol, okay? That's a sterol. Okay, so outer membranes, let's go over this part of it. So gram positive, no outer membrane. I'm just putting this in here so that we don't have any question about that. There's no outer membrane in a gram positive. In a gram negative, we do have an outer membrane. There's more information about it than just that, though. Oh my God, those individually move. Oh. So gram negatives, they do have an outer membrane. All right. That outer membrane . It's just another lipid bilayer. In case you aren't following what I'm typing, that outer membrane on the gram negatives is made up of, it's a lipid bilayer, mostly of phospholipids, just like any other lipid bilayer, like we've been learning about the cells, right? Those phospholipids with that negatively charged phosphate head and the hydrophobic tails, right? So it's a lipid bilayer. And it's going to be studied with proteins. And these serve as channels or as docking points to enter the cells. This could be for other molecules that need to get in or out, or attachment points for the bacteria, and all sorts of stuff that the bacteria could interact with. Also, that can serve as recognition points for invading phages. So for invading phages to attach to recognize their host. Right, okay, so that's basically what that outer membrane is. It does all the work that any other membrane on the outside of something would do, like our own membrane. It's just we don't have two, they have one on the outside. They also have unique lipids on the surface of it. And this can include this includes lipopolysaccharide, which we call LPS, or endotoxin for our purposes. This is a sugar lipid. And our immune system okay, so lipopolysaccharide, this doesn't stretch out. Okay, I guess, I'm just gonna redraw this. I need the space, I apologize. So lipopolysaccharide Anyways, lipopolysaccharide, we abbreviate that as LPS. There's a part of LPS, but we in our class are just going to say LPS in general, is endotoxin. Lipopolysaccharide, LPS, endotoxin, all the same thing. These are all these are all a lipid found on the outside of the gram-negative outer membrane. It's a lipid sugar combo on the outside of the gram-negative outer membrane that our body can automatically see as bad guy, foreign, shouldn't be here. And because of it being able to see it that way, our body even recognizes it that way as a PAMP, so a pathogen associated molecular pattern, which we learned about in chapter 15. It's that bad. It can also serve as a pyrogen. Okay. Which induces fever. Okay, so PAMP, pathogen associated molecular pattern. Pyrogen associated with causing a fever. So that is the breakdown of that. Okay. So I'm going to take another little arrow and go to the cell on the. That's fine, whatever. That's close enough. Okay. All right, we did the gram stain. We told something about it getting trapped in the peptidoglycan. So adding some of these details in. So the primary stain in the gram stain is crystal violet. And then our mordant is iodine. And in our gram negative, safranin is our counter stain. So ethanol is our decolorizer, and our counter stain is so our counter stain is safranin, right? So you guys know the drill with that. It's going to be, let's say I have a stepwise thing. Crystal violet, here we go. Crystal violet, then the iodine mordant to fix the crystal violet in place, then the alcohol to rinse out any excess crystal violet, and then the saccharin to counterstain if the crystal violet got all rinsed out. In the case of a negative, that would be the case. So yeah, so we have that. And here is a little stepwise guide for that one. No, that's still not ready. It's like that. Close. Actually, just go ahead. I don't know why I'm fighting this so much. I have a favor. And just put this here so that you guys have it. And that way you can see the steps and remind yourselves of it here. Just take it out. That'll be better. Put this one back, just go. Okay, that's better. So there's that. We have our crystal violet here. Our crystal, our gram stain here, so you guys can go over that. That's there, that's available. And then endospores, dominant structures, twisting harsh conditions. So let's go ahead and add another page here. So we will come back to that. This page. Alrighty, endospores. So endospores are dormant. And endospores. They're dormant, okay? And this is what Bacillus or Clostridium species, or very, very, very closely related species. Usually they used to be Bacillus or Clostridium. For example, C. diff, I always call it Clostridium difficile. It's now Clostridioides difficile because they renamed it, but it's still a Clostridium. It still falls in that group. Bacillus Bacillus, there used to be a Bacillus spiricus. Now there is a Lyscinobacillus spiricus, but it's still a bacillus and it still makes endospores. But they always are going to fall within these two categories. Acid fast is going to be Mycobacterium. The ones without a cell wall, Mycoplasma. The ones that make endospores, Bacillus or Clostridium. That's it. Okay. If it is Bacillus, it makes endospores. If it is Clostridium, it makes endospores. Okay. So just think of it that way. These two, the Bacillus and the Clostridium, also happen to be gram positive. Okay. All right. When we say dormant, it is no metabolic activity. All right. So dormant means no metabolic activity of any kind, okay? It means not doing anything. Dormant means it's doing nothing. So whenever you have questions on the back of things like your lab question things, and you guys are asking me, so it's endospores, that means that, I mean, it has flagella, I mean, like it's gonna be motile, right? Absolutely not, guys. It doesn't have any metabolic activity. How can it move? Does moving not require energy? I'm pretty sure you guys know moving requires energy. When you want to go get up to go to the bathroom, you probably sometimes think about how much energy that takes you. That's literal energy that's costing you. You have to be using energy to do anything ever in the entire history of all of chemistry. So you're not going to tell me that your dormant endospore is motile, zero on that one, right? It can't be, impossible. That's the whole definition of what dormant means, doesn't do anything. Don't forget about those little kind of details that still apply, like logic wise, on some of these things that, you know, dormant, I mean, it means dormant. It's like hibernating. Is a hibernating bear who's actually hibernating about to go out and run a marathon? No. So don't get these weird ideas just because it's new to you. So dormant, they're not doing anything. What else did I want to say about these? Do I want to go through these steps of it? Yes. If you were vegetative, you're going to be reproducing, you're going to be eating, you're going to be making energy, you're going to be dividing, all of this stuff. So we could call this the normal stuff. If you are a vegetative cell, you're a vegetative cell and you're getting ready to make endospores because, I don't know, things are getting slim. You don't have as much food or whatever to make more energy. When that vegetative cell starts to make an endospore, it then becomes the sporangium, which is the spore forming cell. Okay. That means it's then going under the process of sporulation. And sporulation is the process of making an endospore. Don't read too much into it. Okay, so now let's say finally, when an endospore is ready to become vegetative again, wake up and become vegetative. It's going to go through germination. And that's just that waking up process of becoming a vegetative cell and having previously been an endospore. I think I have a slide for this, but I'm gonna come back to that in just a second. Back up here to one just really quick. We said bacteria and archaea lack membrane structures, everything. So this is us talking an awful lot about how they are basically the same thing. We would consider like the bacteria to be basically all over in our everyday and everywhere lives, right? And archaea are our extremophiles. Who live in extreme environments. Like high salt, high temperature, extremely low temperature. Those very, very extreme niches, that's where the archaea can be found. So I'm going to go over this one because that's a term I know we already know what that word is, but I'm going to bold it just in case we forget. Pleomorphic and staphylo and strepto and palisade. And definitely those are in the same one. Tetra and stuff. I remember I'm probably which are fine. All right, there's that, there's that, there's that, there's that. This seems bigger than the others, and I don't like that. I think it's just because it's sued. If you're trying to tell the difference between the identity of things, one of the manuals that we use is called Berge's manual. That's all I need you to know about that. It's called Berge's manual. And you can go to it. There's physical forms as well as online forms. And you can like look up basic information based on color changes of media or genetic information, all sorts of stuff to determine what your organism is using Berge's manual. Okay. So I will just, I guess put that to Berge's manual is used to identify bacteria based on physiological, immunological, and genetic tests, among others. There's all sorts of tests in there that we can base off of, but it doesn't have to be our basic categorized situations like we've had before. So that, I'm just going to leave that one there. And I think how this is scrolling. Why? I didn't even click on it. And then lastly, you can I would put this back on page 1 when we were talking about the cell envelope, but I just don't really have, well, we can maybe fit it. We can maybe fit it on there. So it's all in the same space. So there's 4. So we're gonna start off with just the descriptions first. So you could have guys that are gram positive. You could have guys that are gram negative. You could have guys that have no cell wall. Or we call those guys mycoplasma, excuse me. No, it's not big. I'm sorry, I have like number buttons on the side of my mouse, and sometimes I click them. And then our last one could be, you could be archaea. They don't even have the same stuff that makes up their cell wall. They don't have peptidoglycan. They have these weird hydrocarbon. And when I say hydrocarbon, I'm not talking about like, oh yeah, we know hydrophobic tails. No, 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 no. I'm talking about things like octane and stuff. Like they have like hydrocarbons that are not like what we would consider like cell-based molecules. They have some weird stuff going on. The archaea do, but whatever. So they have their own business. So if you are gram positive, the term that the category that you fall into is firmacute. These are the firmacutes. Okay, so I remember that because gram positive have the thick cell wall makes them firm. So that's how I remember that. That's not like an official thing, obviously. That's just how I remember. All right. Let's go to the next one. I go through these systematically when I try to remember them. So I'm going through my system. So I either start with the gram positive or I start with archaea. For me, archaea, these are the Mendozicutes. And I remember this because my old boyfriend, I think he was my first boyfriend. His name was Joey. And he was cute. So we'll say that. We will even say old, which to me is similar word to archaic. Okay. So lucky you guys that you get to have multiple choice on these. So that kind of helps you at least remember based on that. But there's 2 more left. There's gracilicutes and there is tenericutes. So I'm actually going to go down here really quick and just write those down. Gracilicutes and tenericutes. Okay, so this is how I remember this, okay? I will go to my 2 choices, gracilicutes and tenericutes, and I will say to myself, gracilicutes, that starts with G, like gram negative. So obviously no cell wall, that doesn't have a G in it. So that is literally what I do for the last 2. I'll say gram negative, that's G for gram, G for gracillicute. You can't do it first, right? You have to knock out gram positive because you had to get rid of the firmicutes, and that that thick cell wall, gram positive. So you have to get rid of those two first, the gram positive and the archaea. But now all we have left is gracillicutes and tenericutes. So I will put gracillicutes here. And that will be because that G gracillicutes matches to the G in gram negative. Okay, and then we have the no cell wall group, and so that leaves tenericutes. So we will go ahead and say, after we add gracillicutes, and you can even just try to, like, you'll have the multiple choice, but hey, at least here you can have gone through it. So, I'm literally just gonna put my note first, but that's tenericutes is left over. Okay, and then goes that. I would ask you guys if you have any questions, but there's nobody on here. So no biggie there. I wish there was a way to. Is there not a color? I can't shoot a color for this. Well, Bob. Sorry, I'm trying to figure stuff out. I'm wasting your time on that. But there we go. So that one's in. Okay. I think that's it. Sorry, let me check my. That's good. Okay. So that's that for that part. So now I am going to go over some critical thinking open-ended questions that are relating to this chapter. I know this is going to be a little long, but I'd rather it be long and cover, the gist of everything, and then be able to let you guys go over this on more free time and have everything that you need. Just some more figures to this study session. Let's see. We got the other one and the other images. Yeah, that'll be better. Okay, cool. Okay, so I'm pretty happy with that, and that works for you guys too, as far as information to help study for chapter 4. I'm imagining there's not a whole lot of, right now anyways, desire to go over chapter 4. People think there isn't anyways, and I'm betting that when they get into studying, I'm hoping that at least some of you will be able to come back to this as a good resource to study on. So having said that, there is the whiteboard for this one. And so I'm going to jump right into our chapter 4 study session open-ended questions. Let's see, let me share my screen. All right. See, like you can literally go away. Okay, so our first question, define the term ubiquitous and explain whether this term can be used to appropriately describe bacteria and archaea. I want you guys to be clear that like on some of these questions, especially this one, and ones we might see in the future in some of these other study sessions, this doesn't give, and I don't give a crap about whether you understand the word ubiquitous. You're not going to get tested really on what ubiquitous means. That's not the point here. The point here is for you to recognize the fact, number one, that ubiquitous means everywhere. Like it is just pretty much anywhere you look and test, you could find this, that, or the other thing. That's what ubiquitous means. That's all this is asking, that you be aware of, that you know what this term means, so that when we go to apply it to bacteria and archaea, can you explain how that would work? So this is one of these moments where like, I'm going to post this PowerPoint online on Canvas, and I would hope that you guys would try to go through these questions on your own, take it through your own time. I'm not going to answer them and then go through the answer. What I am going to do here as I'm going through these questions is I will ask the question, maybe post some thoughtful information, and then post the answer and go over why that's the answer. So that's what you can expect here. But I highly encourage you guys, when you get to this part, just open up the PowerPoint presentation and just go through it yourself without looking ahead, and try to see if you can go through it or figure out what the answers might be or how you might answer it if you were posed with this question, okay? So that's the idea, to get you thinking about the stuff in this chapter. So define the term ubiquitous. So I am just going to tell you right now, that just means everywhere, all over the place, like anywhere you could possibly look, they're found pretty much every single where. And can that be used appropriately to describe bacteria and archaea? So ubiquitous, it's everywhere. It is appropriate for describing both bacteria and archaea. They have been found colonizing pretty much every habitat known to man, excuse me. Archaea have been found in the most diverse environments, leading to the use of the term extremophiles, which we actually already applied in our study session to describe these organisms. So yes, ubiquitous could be used to describe both bacteria and archaea. Number 2, quorum sensing is a process used by many bacteria for communication. It involves the production of molecules called autoinducers, which act as bacterial chemoattractants. Describe how a motile bacterium would use its flagellum to respond to such a stimulus in its environment. This has a lot going on in this question. We have talk of quorum sensing, which is a basis of communication typically within a biofilm. That's how we've learned about it, but it doesn't have to just be in a biofilm. It's in any environment where there are other bacteria, and there can be production of autoinducers or any sort of chemical that can have an effect on nearby bacteria, telling them to go to or away from something in response to those chemicals, right? So how would we use our flagella to respond to chemical signals in our environment? We literally just talked about chemotaxis, that is responding to chemoattractant or chemorepellent, which would be something like an autoinducer in any sort of environment where bacteria can be communicating to one another via quorum sensing, whether that's inside of a biofilm or outside of a biofilm, it's irrelevant. So a flagellum rotates counterclockwise, cell itself swims in a smooth linear direction towards the stimulus. That action is called a run. Runs are interrupted by intervals of tumbles during which the direction reverses. That rotational direction of the flagella will, like if you're just going like this, all of a sudden back the other way, and that will stop and change our course. And we can alternate between running and tumbling to generate what they call a random walk, and use more runs or more tumbles to bias the directional movement, basically, is what this is going on for. In response to stimulant, attractant, let's say, that's what I need to say, in response to an attractant or to get away from a repellent via chemotaxis. All right. Number 3, based upon your knowledge of cell wall structure, explain how microbes causing meningitis and typhoid fever can induce fever and systemic shock in an infected patient. This is an interesting question, especially since it applies pretty directly to the type of stuff that we're going to be going over in our upcoming chapters in Unit 4. So let's just break right into this, okay? So we know about cell wall structure. We learned about that outer membrane of the gram negatives. So this is really what we should be thinking about here. So I'm going to tell you this. A gram-negative bacterium called Neisseria meningitidis is the meningococcus. The meningococcus is the most deadly cause of meningitis. So it is a diplococcus. It's round and it comes with like little butts all over the place, but they're really quite cute. But the Neisseria meningitidis, that one is the most deadly form of meningitis you can get, and it is gram-negative. So it does have endotoxin on the outside of that outer membrane of the gram-negative envelope, right? The cell envelope. And then typhoid fever. Typhoid fever is caused by salmonella. It's a certain strain of salmonella, but again, gram-negative. So here we're talking about inducing fever because endotoxin, or LPS, lipopolysaccharide, found in the outside membrane of gram-negative bacteria, is a pyrogen, so it induces fever just naturally by existing there, and can induce shock due to its appearance, because there shouldn't be any endotoxin in your bloodstream, and your body will respond in a sort of panicked manner, and that will induce all sorts of like a chemokine response, inflammatory response, and all that sort of stuff in response to the presence of, I'm just answering the question at this point, but bacteria that cause these diseases are typically gram-negative. They have a thin peptidoglycan layer. Okay, cool. That's why they test gram-negative. And then an outer lipid-based membrane that has lipopolysaccharide in it, which is endotoxin. That can trigger systemic shock in infected patients, as well as act as a pyrogen that causes fever. Number 4, provide evidence in support of or refuting the following statement. The cell or the cytoplasmic membrane, the cell membrane or the cytoplasmic membrane or the inner membrane is a non-essential structure in bacteria because its function is replaced by the cell wall in these microbes. I mean, this is setting you up for failure here. So this statement is false. This is because that cell wall deficient bacteria exists. We've already talked about them, right? The mycoplasma bacteria, they don't have any cell wall. All they have to rely on is the cytoplasmic membrane. So they need that in order to contain everything within the cell. So mycoplasmas rely on the cell membrane to resist lysis. They also have the sterols to help with cell shape, which isn't very specific for these guys. But in addition, the cytoplasmic membrane that contains everything within the cell itself carries out important functions such as energy reactions and nutrient processing, and thus plays an essential role. And that's something we didn't talk about in this chapter, but we did get into in like chapter 10. All of the energy making processes of the bacteria, because our cells, which we learned about primarily when we were talking about this stuff, was our cells undergo aerobic respiration, to make our ATP within our mitochondria, in the folds of the mitochondrial membrane, right? But that's nice. But bacteria do not have membrane-bound organelles. They don't have mitochondria. So they do all of that. They still need energy, just like the rest of everybody else. So they do all of that on their inside membrane, that inner membrane, that cytoplasmic membrane. So it is essential to the survival of that organism. This stuff cannot happen on a cell wall. It can't. It's impossible, chemically speaking. So we have to have the inner membrane. It's not superfluous. It's not a useless artifact. It is absolutely essential to the survival of the bacterial cell. Tornado sirens. It's just a test. In case you were worried, I know you weren't. That's okay. Right. So open-ended question number 5. Describe the characteristics of an endospore-producing bacterium that make it an ideal candidate for bioterrorism, but an undesirable intruder in a hospital setting. And we've mentioned this multiple times. You guys are probably sick of hearing me talk about anthrax and C. diff. Yeah. And then explain why the production of endospores is not considered a method of reproduction in most bacterial species. Not a method of reproduction. I want to add that. The production of endospores is not a method of reproduction. It's a method of preserving bacterial genetics, waiting for a better condition so that you can reproduce with your vegetative cells. The endospores are not a reproduction means. It's a life-saving, life-preserving means so that later, whenever there are better conditions, then you can reproduce and be a real cell. So endospores are hardy, resistant structures, making them ideal for dispersal and survival under numerous conditions. This can be beneficial for bioterrorism because we can release it as a weapon or an aerosol powder form, anything like that, because they can withstand being dried out and then redispersed into what might otherwise be considered potentially harsh conditions. Now you're putting endospores into conditions that they can withstand just fine because they're already endospores. So that is useful for bioterrorism, unfortunately, very realistically so, and has already been applied in many situations because of that. It is a deleterious agent in, I guess, situation in clinical settings because endospores are resistant to routine cleaning chemicals. It's pretty hard to get rid of them. Most chemicals that you are using to clean surfaces aren't sterilizing agents. They aren't sporicidal. So remember, sporicidal is any sort of an agent that can kill endospores specifically. And so you want sporicidal agents in hospital settings because we're trying to eliminate spores like C. diff and stuff like that, right? Endospore production is not considered a reproduction because in most bacteria, only one endospore is produced from every vegetative cell. So one cell becomes one endospore. Vegetative cell goes through, sporulation. Sporangium makes one endospore, and through sporulation, and then you have an endospore that's waiting to germinate and become a vegetative cell. But that's just 1-to-1 ratio every time you go through it. Only the vegetative cell in a healthy, normal, metabolically active state can make new cells, not the endospore. So it's not an actual reproductive mechanism. It's just a way to survive harsh conditions. All right, that is that, and that's everything that I have. Wow, it got dark. Thank you, guys, for, I would say, joining. Nobody did. That's not what I mean. I'm just saying, thank you guys for watching this later. Hopefully you watched this later. I wasn't mad about you. I just want to be clear. I wasn't mad about people not showing up. I was just mad about my freaking computer not working. That was really annoying in the beginning, in case you couldn't tell. So, annoying computer, and I don't understand it still, but that's all right. I'll figure it out at some point. So, thanks for coming and watching this. If you did, I hope this helps some of you out. And let me know if you guys have any questions as you are studying, and I will see you all next time for chapter 5, which will be the eukaryotes, which I think more people need more help with that anyways. So that'll be on Wednesday. That'll be in person and on Zoom, pretty much the same way. I'm just basically doing it in the lab instead of at home. So it'll be the same, but I'll see you guys next time.